Welcome, listeners, to Talking Eyes. I'm your host, Leanne Trin, an optometrist and public health clinician based in Larrakia Country, also known as Darwin in the Northern Territory of Australia. This podcast was created to translate exciting, innovative developments from vision research into a form that both clinicians and really any curious mind can understand. Innovation, simplicity, clarity. This series will be diving into a topic where the research has been absolutely awe-inspiring: the retina. It's just a small part of the human eye, roughly the size of a postage stamp, but it's an incredible piece of human biology that plays a central role in how we experience the world around us. There was once a time we accepted the fact that retinal cells, and indeed all neural cells, once damaged would be lost forever. But imagine if damaged cells could be replaced and any vision lost could be regained. In fact, what if we could replace the entire retina? Stem cell technology is mind-blowing. It's the next exciting frontier in vision research and could essentially be the cure to everything. This episode will delve into the science of how stem cells work, the exciting progress being made in clinical trials, and the real-world impact this technology could have on people who have lost their sight, and even those born without sight. We'll share stories of breakthrough treatments and discuss the potential and the challenges of bringing these therapies from the lab to the clinic. I had the absolute pleasure of chatting with Dr. Anaya Gonzalez-Cordero, the research lead in stem cells at the Children's Medical Research Institute in Sydney.
Hello, good to be here.
Did you dream you would be working in this sort of field when you were a child, Anaya?
Not really. I knew, I guess I knew very early on my studies that I wanted to be a scientist, that I wanted to do research, but I didn't know I was going to be studying the eye specifically. And it was actually a very important decision in my life when I had to make the decision, okay, I'll dedicate my career to study eye and try to develop therapies for blindness. So I was actually in university in Brazil doing biological sciences and just studying all the different types of biology. And I decided that I wanted to go and learn English in the UK because I needed that to study science. So I was supposed to be there for 6 months, but I stayed for 20 years. So I stopped my biology course in Brazil. And after a few years in the UK, I said, okay, let me go back to university. And that's when I applied for University College London, UCL. And I started my degree in developmental biology. So it was great because I knew I wanted to be, it was more of a narrow degree in terms of development of the embryo. And that's where perhaps my passion for stem cells started, really. And I did my degree and then moved towards my PhD, and I was very lucky to get a Wellcome Trust PhD scholarship. And in this scholarship, you can experiment in 3 different labs on your first year of research. So I did 3 completely different things. One was studying brain development in zebrafish. The other one was cardiac development in stem cells in mouse. And the other one was developing stem cell therapies for the eye in the Institute of Ophthalmology.
3 completely different areas.
Yes.
They couldn't be more different.
Exactly. And then, you spend 3 to 4 months in each of these labs. And at the end of the first year, you have to make a decision.
Zebrafish,
where I'm going to spend 3 years of my life, studying and dedicating my life completely to it. And then you stop and you think, okay, do I want to study this? Because then once you do your PhD, of course, you can change pathways and study other things later during your postdoc. But it's a big commitment. And I just make a list of pros and cons for each lab, and I decided, there I go into
We're
that.
so lucky to have you. Let's go to stem cells.
Yes, so the textbook definition of stem cells are that they are these, I like to call them ground cells, so the basic material of the body, right? The building blocks that they self-renew, so they proliferate, no, they don't stop proliferating, and give rise to another population of cells that it looks just like them. So it's just that self-renew population of cells. And they also have this amazing capacity of differentiate. And when I say differentiate, is the generation of a new cell type. So now they are going to proliferate and generate a new stem cell that looks exactly like the mother, but also a cell type, a different cell type. And that different cell type can be any cell in the body.
And so, do we have stem cells in the eye? Are they everywhere in the body at the moment, even beyond birth, beyond growth? Because I imagine they would be everywhere when we're babies, when we're growing, because obviously we
Yes.
need to duplicate the cells so that we get bigger, but also change the type of cells because we need cells of different types. So, are they just all over our bodies, and are they all over our bodies for life?
So we have different types of stem cells. The ones that you are talking about are what we call adult stem cells. So they reside in specific tissues in your body. So, for example, your skin, your lung, your blood, and they are a population of stem cells that are multipotent. So they can give rise to new cell types and also self-renew, but only cell types of that specific tissue. So, as you know, for example, your skin is replaced on a daily basis, and there are stem cells that reside on your skin that will be the ones replacing the epithelium, the different cell types of the skin. So those are more constrict in their capacity, let's say, right? And they are the majority of our organs. Now, and I'll come back to the adult stem cells in the eye. But before I go there, maybe I need to introduce the embryonic stem cells and the induced pluripotent stem cells. So the embryonic stem cells are the pure, the master stem cells of our body. So they are localized in the very early embryo. That's why they're embryonic
They're the
stem cells. And
originals.
it's a small population that will give rise to your body, right? To all the germ layers, to all the cell types, organs, and tissues, and so on. So they are pluripotent because they have this multitude of this capacity, this open capacity to generate different cell types. Now, induced pluripotent stem cells are very similar to embryonic stem cells, but they have been induced to become stem cells.
Right.
They can give rise to all the different cell types of the body, so they're pluripotent, right? Just an embryonic stem cell would be. And in fact, when you look down the microscope, they are exactly the same. But the beauty here is that we started the generation of an induced pluripotent stem cell not in the embryo, but with a cell that was extracted from an adult body or from a child. So a cell that is already committed.
Right. So one
So
that had
let's say
differentiated already into a very
it was
particular
already,
cell type, and then you've changed the cell somehow so that it can now become whatever cell type you'd like it to differentiate
yes,
into.
it goes back. We induce this cell back. So that's why they are
Yes.
induced. We induce them back into the stem cell. So let's say in the lab, we work a lot with blood samples and skin samples. So you can isolate a vial of blood from a patient or a healthy individual, and we will separate in the lab a specific type of the cells in the body and in the blood. And then we use these blood cells to then introduce 4 different transcription factors. So these are proteins, so just some molecules that are essential for stem cell development. So we know that these 4 factors are important for stem cell development, normal stem cell development. We artificially introduce this into the blood cells. And then after a few weeks, these factors will get expressed inside the cell. They'll be on, so they'll be turned on. And after a few weeks now, we start seeing the blood cells that were floating in the dish. They start to attach in the dish and form colonies that look just like stem
Wow.
cells, as they should look, as the embryonic stem cells
Yeah.
look like. So now what we did, just by providing these four factors, we turned the blood into a
Wow,
stem cell.
that's incredible. Anai has introduced some new and complex concepts so far, so let's recap some of them. There are different types of stem cells, and the ones from the embryo, the embryonic stem cells, are known as the original master builders. They're called pluripotent because they have the capacity to transform into any type of cell in the body. Now, these embryonic stem cells were first isolated from mice in 1981, and then in 1998, we had the first documented isolation and culture of human embryonic stem cells in the laboratory. It took almost 2 decades because of the legal, ethical, and social controversies surrounding this sort of research. Isolating stem cells from the human embryo was essentially seen as being equivalent to destroying a human life, because when stem cells are isolated, the embryo does not continue to develop. Now, in 2006, a person from Japan, Shinya Yamanaka, made the remarkable discovery that by introducing 4 genes, often referred to as 4 factors, into a mature differentiated cell, it could be reprogrammed or induced back into a cell that was pluripotent, what we call induced pluripotent stem cells or iPS cells.
A major part of the benefits of using iPS cells, it's because we don't need to use embryonic stem cells anymore. But perhaps the most relevant part for us studying diseases is that same genetic material of a patient. So now what happens is that we have a patient with an inherited retinal degeneration, Stargardt's. We can isolate the blood from this patient. We take it back to the lab. We do the reprogramming. So this generation of blood into iPSCs is called reprogramming. So we do our reprogramming and we generate now stem cells from this Stargardt patient. Of course, because the blood, we have the same genetic material in every single cell in our body. Now the stem cell has the Stargardt mutation.
So would you be able to treat somebody with Stargardt's with somebody else's stem cells? Does that make sense?
Yeah.
So you're removing, say I have Stargardt's, you've removed my cells, created these iPS cells. Could you use those iPS cells to treat a different person with Stargardt's? Because essentially they're my stem cells.
Yes. That's perhaps another concept that I need to introduce here, is that so, for example, with adult stem cells, and one example of adult stem cells are mesenchymal stem cells. So people might have heard of that. So, cells that can give rise to cartilage, blood, and fat. These cells, they have neurotrophic factors. They express factors that can be beneficial to other cells in the body. And they are used in therapies. There's some approved, there's some that are in trial for neurodegenerations, for example, because of this neurotrophic effect that they will provide to the environment, right? Those are stem cells that are used, transplanted into the body and used for potential treatments. Now, if you're working with induced pluripotent stem cells or embryonic stem cells, you wouldn't necessarily, and I wouldn't recommend it, that you transplant and use those stem cells in any part of a patient of the body, because these cells, again, they're proliferate, they're highly proliferative. They will form tumors and they're pluripotent. While the mesenchymal stem cells, they are more defined, it's a specific population. With the embryonic stem cells, because they proliferate and they have this capacity, they will form masses and they will. So we're not
doing that. That's not what
please,
we're doing.
we're not doing that.
But you've grown cultures of healthy photoreceptor cells in a dish in the lab.
Yes.
and then use the cells to replace defective cells and restore sight in laboratory models of hereditary blindness. Can you talk us through that work that you've been doing?
Yes. So that's the difference there. Instead of using stem cells, what we do is to now use the stem cells and differentiate them. So we inform these stem cells based on developmental biology, and we know how to do that. Okay, go on and form an eye, become a retinal cell. Become a retinal progenitor cell, and then you can go on and mature and form a ganglion cell, a photoreceptor cell, any other retinal cell type that we
And
want.
we know that those cells won't self-replicate again. And so you've created these little organoids.
So the field started with this differentiation of stem cells into cells in the dish, using basically standard culture conditions, a monolayer of cells that grow a little bit organized, but really not much of an organization. So we had a bunch of photoreceptors here, a bunch of retinal ganglion cells there, but not a lot of organization really. It was fantastic. I have to say, first time I saw these cells in the dish, I was like, wow, we're generating these cells from scratch, from stem cells. But a landmark study from Japan, actually in 2011, demonstrated that now if you put the stem cells in suspension, forming a ball basically so you just form a ball of these stem cells with certain number of cells, and you add a matrix around these cells in suspension. They will self-organize and form a mini retina in the dish. So spontaneously, this extends, of course, we will provide growth factors and media formulation that will induce these cells to become a retina as opposed to a heart, for example. But they spontaneously can form retinal cells, and this and the lamination of the retina, and these organoids now have all the different cell types of the retina, from ganglion cells to photoreceptor cells, RPE cells. And we started the field with mouse embryonic stem cells. So we generated mouse mini retinas, and then we moved on into human, and now we have human organoids growing in the dish. So we have the stem cells. We can have healthy stem cells from healthy individuals, and we can have disease stem cells that we generated from patients, right? Then we grow the stem cells, we generate the retinal organoids, so beautiful laminated structures with all the cell types. And now we can study what are the difference between the healthy and the diseased in the dish, because we have a number of these organoids growing, right? So now I can say what is different in the photoreceptor cells from a Stargardt's patient when I compare with a healthy individual. So that we call disease modeling. So we're modeling disease in the dish. And I can do that for a number of Stargardt's patients. And even though it's a heterogeneous disease, we should see the same phenotype. So there's something that is leading. Of course, we know the mutation, and we understand what is the Stargardt's is causing to the ABCA4 gene, for example, and the protein. But there are general disease phenotypes that we don't understand, and now with the human cells, we can start understanding. And then, because we have the organoids growing and we have thousands of these organoids growing, we can test, okay, is this new drug going to rescue the photoreceptor phenotype? Is this gene therapy going to be efficacious when I treat my retinal organoids? So I can test new therapies in vitro in a high throughput manner.
So you don't even need patients then, do you?
I just need to have a good representation.
You just need a few of their cells. And then you can create basically a replica of what their retina is or is doing, and then compare that to the normal and sort of try to define treatments for it without treating the actual patient. That's incredible.
Yes, and another really big branch of my lab, it's looking into improving the organoids. you
Yeah.
know, because the technology is relatively new and is growing, so we want to have better models. uh, for example, one of the recent discoveries that we did is that we, just by changing the media formulation and giving more lipids and glucose to the media that we give to
Mm.
these organoids every day, so it's their
Mhm.
food, we could create more outer segments. So our photoreceptors before had rudimentary outer
Mhm.
segments, which was good. And we were like, wow,
Amazing.
yes,
Yep.
we're generating outer segments, which is already incredible. But now, by providing lipids, which is, of course, what the outer
Made of.
segments are made of, we have nice disc-like membranous outer segment. Another thing that we're doing, we're combining retinal organoids with brain organoids, because our retinal ganglion cells are in the middle of the organoid, and they end up dying with time in culture. So, as I said, these cultures go over months, because we do respect the development of the eye. So the ganglion cells, they end up dying in the middle, and we don't know if it's because they're just not receiving enough food, enough oxygenation, or it's because they're lacking their connectivity with the brain. And of course, then we have almost like a glaucoma model, that where your retinal ganglion cells are dying. So what we did was to connect the retina with a brain organoid, and what we see is that the ganglion cells and the retinal organoid are now connecting, sending axonal projections towards the brain and populating the brain
So it's
organoid.
really
So we
simulating
think,
the, what is actually happening in real life. And I was going to ask actually, because visual perception is much more than just a piece of tissue. And we know with development, we know in children, when they're born, they actually need visual stimuli in order to develop their visual perception. So this must be such a challenge for you to replicate in a lab setting.
yeah. Yeah, and I have to say that until very recently, it's overlooked. It's overlooked in a lot of cultural conditions, because all we do is really change media
Yes,
and give new
provide
food
the
and add
ingredients,
more
but not the
factors.
actual stimuli
Yes,
that.
we don't provide that. So when I moved to Australia, actually 5 years ago now, I decided, okay, I had this idea, and this was more related with cell transplantation, which we haven't even touched yet, which is a whole new area that we do in the lab. I decided that we were going to introduce electrical stimulation into our culture conditions, because neurons normally have these, all the cells, not just neurons,
Mm.
but all the cells have electrical fields, and they use that to localize the receptors for calcium. For correct calcium input and output. So we said, okay, we're missing this in vitro. Let's start a program of research that delivers electrical stimulation to the organoid. So we saw increase in proliferation. We saw increase in photoreceptors yield if we deliver electrical stimulation. Quite a few changes
Mm.
happen once we
That's
start.
exciting.
And now, of course, the next one that I really want to do, and hopefully we can start with a PhD student doing that soon, is light stimulation. Because, of course, for the retina, that's the input that we
That's the
need.
main ingredient, isn't it?
Yeah.
So, cell transplantation, let's talk about that, because that is where things are at for vision regeneration, vision renewal in some cases, and potentially will be part of clinical practice in the future. So, what does that mean?
So cell transplantation, cell therapy, it's now replacing the cells that were lost during disease and blindness.
So these are
So
for conditions
while
like retinitis pigmentosa, where there's cell atrophy, geographic atrophy, and macular degeneration, perhaps. Yep.
Yes, yes. So we call it a more of a universal treatment, because at the end of the day, all these conditions will lead to some kind of atrophy and loss of cells,
So this is
right?
the stage that you're
So
looking at entering with stem cell
more
technology.
advanced
Yeah.
stage where the cells have been lost. Yeah. Of course, in the future, if you think about when this is all working and then we need to optimize, hopefully one day it will work. And then at that stage of optimization, one thing that you can go earlier so that perhaps the endogenous cells will not suffer as much because they have the other healthy cells around them. Or you don't necessarily need to treat an advanced retina, diseased retina, where perhaps your other cells there, the interneurons, are not as healthy anymore. And so there will be advance, of course, but right now we are targeting the replacement of, and I work specifically with photoreceptor cells, so the replacement of photoreceptor cells at advanced inherited retinal degenerations when the cells are no longer there. When the cells are still there and it's an appropriate target, we can do gene therapy, for example. So for gene therapy, you do need the cells to be there in the first place to receive the viral vector. For cell therapy, in my opinion, what we need to do is to make good new photoreceptor cells and transplant these into the eye in the hope that they will make new connections and then have respond
And
to
have a
light.
viable, functioning retina. And so these clinical trials that you've talked about where there has been therapy and implanting these cells essentially, the goal of these trials have been to, have they been to restore vision?
Safety mainly, so they're phase 1 and 2 trials. So it's mainly to show that these. So in the first one, for example, it was retinal progenitor cells isolated from fetal human retinas, and it's just to show that they're safe, and they've shown that, and a moderate effect on the endogenous retina.
So sort
For
of
example,
just the
not necessarily
physical, it's safe, it works, it fits in, it's there, and it's not causing any harm.
to a certain extent.
Yeah.
Exactly. Exactly. There's no rejection. The cells will be there surviving for a certain period of time, and that's what happens with all these initial
Yeah.
trials, right? We had a few trials with replacing RPE cells, for example.
Yeah. Something that Penny Allen mentioned in her episode on the bionic eye was what the tissues are like at sort of the end stage of diseases. The vascular tissue is a lot thinner. And so is it sufficient to be able to provide nourishment to new stem cells? Will the metabolism of the stem cells drive the vascular system into activity again? Do we know how that works?
Not really. I would say the field, it's in its infancy still. What we assume is that the interneurons, we know that it's a degenerated
Yes.
retina, right? So we assume that the interneurons are still receptive and they're still healthy. But in AMD, for example, we know that the Brooks membrane it's damaged, that the vasculature is not good. So in those extreme cases, replacing RP alone might not help. Might be that you need a sandwich of something that is choroid like RP with a Brooks membrane, and that might be a better
Implant than just
transplantation.
receptors.
Yeah, but we have to start. So the field started with RPE cells because RPE cells were easier to generate from the pluripotent stem cells. So both from embryonic stem cells and iPSCs. It's a population of cell that is the differentiation protocol is relatively easy and quicker if you compare with retinal organoids. They form a nice monolayer of cells just like they would do in the eye, and you can transplant these cells as a patch. So it evolved much quicker, and clinical trials were done earlier and shown some efficacy. For photoreceptor cells, because it's harder to get these cells, it's taking a bit longer. But in fact, we have a trial ongoing in Japan where 2 patients have been injected with a piece of the retinal organoid. So they developed the retinal organoid, they cut what they call a sheet of tissue, and they transplant that into patients. They did a lot of preclinical work as well, while my work transplants a purified population of photoreceptor cells. They chose to go with the piece of the retinal organoid intact. And they actually show really good survival and long. I think they are 2 years post trial, and they show survival and some level of efficacy. So just
Efficacy is in terms of vision,
yes,
restoration. Yeah,
yeah,
right.
yes. So it's very exciting, and there are other laboratories developing this therapy. So I'm doing it here in Australia, but there's David Gum doing it in America, Professor Robin Ali doing it in the UK. So I come from Robin's Ali group, and the Japanese, so Masayo Takahashi is the one that is leading this trial in Japan. So it's happening, but it's a bit more tricky because now, of course, we have to transplant photoreceptor cells. They need to connect with the interneurons. They need that environment to be receptive. And they then need to mature to a level where they are light responsive and they can then send signals to the brain. There's a lot of work that we did in the past showing that a specific stage of the development of these cells, so we don't actually want to transplant mature photoreceptors. We want to transplant an intermediate stage where they are already committed to photoreceptor cells, but they're photoreceptor precursors. They're still quite young, so they don't have outer segments yet. So we need to make them form outer segments once they're transplanted. Now, going back to your question of how is the environment of the eye. So, for example, for AMD, the next generation transplantation will be a patch of photoreceptors with RPE and perhaps choroid underneath. We're all working towards now making more complex
Structures.
transplantation cells that can then restore the entire. But it's a stepwise
Yeah.
process, right? So we started with RPE, retinal progenitor cells, RPE, now photoreceptors. There's a lot happening in ganglion cells as well.
And so when we see these technologies being rolled out in clinical practice, will they be disease specific, or do you think they will be general, one solution for multiple diseases?
Yeah, that's a good question because for cell therapy, we can go broader for sure. It doesn't need to be retinitis
Yeah, it's not
pigmentosa
like
or
gene
just
therapy where you have
AMD.
to be very specific
No.
with your genes.
Yes, yes. And will be on us in the field to set up the criteria of who we will include on these trials, and what is the retina, what do we need to be able to include a patient in these trials. Do we need the natural history for certain conditions so that we know they will be perhaps a bit more susceptible to receive these cells and so. But I see it being a more universal. So the Japanese group is targeting retinitis pigmentosa specifically because in retinitis pigmentosa you're losing both your rods and your cones, and they're transplanting a sheet that contains both. But for example, if one is transplanting a purified population of cone cells, which we've done in preclinical studies, we can perhaps target the small area of the macula that is damaged in Stargardt's, for example. So we will be able to tailor because we can generate rods, cones. We can transplant a mix of those. We can transplant just cones. So it will be more
Yeah. And
broad.
so it's literally just ordering online which type of cells you might need for which condition.
Well, I wish it was as easy as that.
Next year, you're hoping that
I really
technology will be rolled out?
yeah. So we in fact we are now moving towards generating. So as I said, I haven't explained the protocol of generating the cells, but we need 3 months, 120 days to generate photoreceptor cells at the stage that we are happy to
Right.
transplant that. And this is a long period. If you are, for us in the lab, we're now used to it and it's fine. The researchers deal with it. But if we are to now manufacture these cells in a good manufacturing practice facility, GMP facility, to be able to use this in humans, it's a long process. So CAR T cells, you generate the cells in 7 to 10 days, for example, and they're ready to go into patients. We need three,
Yeah,
four
wow.
months. And it's a process that is it requires manual work. It requires lots of media change. So there's a lot of components there that needs to be in place, and quality controls for us to manufacture these cells, to even to go into trials. Of course, trials have a less scrutiny, but if we want to make this into a therapy, we need to be thinking about the future, not just trials, but also how we're going to have a product can be commercialized and so on. So it is a long process, and we are working. I have a 5 years study in grant to actually translate this technology step by step into GMP, so that we can perhaps have a therapy. And cryopreservation, how are we going to preserve it and have the vial of purified population cells? How are we going to isolate the photoreceptors out of the retinal organoids? And so there's quite a few questions that we're now trying to
Yeah.
target
So
to.
how far away do you think clinical application will be, considering all these issues? There's so many complexities, not just around the development of the stem cell technology itself, but the implementation of it.
Yes, exactly. So the field moves quite quickly. Again, we already have this 2 patients that received retinal organoid tissues, and it's very exciting. And there's others developing the therapy. But for example, I have 5 years to have that in place, and maybe towards the end of that process, we'll apply for grants to be able to then do manufacturing in an actual GMP facility. And then after that, you have to then do, in parallel, you do large tox studies, and then you have to find the money to be able to do the clinical trials. So we are a few years.
This is research
But
conundrums.
I believe in the technology and the data, the preclinical data. It's very good. So I think it's a question of us, steadily moving towards it. We'll definitely get there.
Thanks for listening to Talking Eyes. Additional resources for this episode can be found in the show notes. If you found the content of our podcast interesting, feel free to post a great review and share it with your community. If you have a topic you'd like to hear more about, or have expertise in the field of vision and would like to contribute to future series, contact me via our socials on Facebook and Instagram. I'm Leanne Trin, your host of Talking Eyes.